Hello, today I'm going to tell you a story about how you can make green gold from plant breeding. But before I begin, let me tell you a bit about myself. So yes, you can tell from that photograph there, I had a lot of hair when I was younger. I went to school in England and I used to play hockey for the school as well as for my university and for clubs. And the picture on the right is me and my wife. She's a professor of genetics at University Malaya. So our kids had the misfortune of being brought up in a house full of geneticists. Right? Now on to things which are a bit more serious. Mankind has a huge looming food problem that needs to be bridged or addressed. If our population is going to hit 10 billion by 2050, we will end up having a large food gap, which is estimated to be about 56% if we have not improved from the 2010 yield levels. And in order to expand land area for agriculture, it will not be possible because huge amounts of land will be required and it will be an unmitigated environmental disaster. So can we do something? And what can we do to address this? So before we start on that, let me take you back and start talking about this molecule that all of you are aware of. This is DNA. And you know that you inherit one strand from your mother and one strand came from your father, right? And there are some features on this strand of DNA that allow us to use those features as tools to look at genetic distances or differences between different individuals. Say, for example, from the difference or the similarity between humans and chimpanzees is 96%. And the similarity between any of us and our siblings is about 99.95%. So a lot of that variation that you see, very small variation that you see between different genomes is all down to single nucleotide polymorphisms or SNPs. These are single base pair changes in the genome and they represent about 1.23% of the human genome. So as scientists, we use these SNPs as a tool, as a cheaper tool, let's say, to study genetic differences rather than the more costly gene sequencing. So why oil palm? Oil palm is the most efficient oil crop by a long way. And these are average yields, global yields, right? But say the best companies are delivering something between 6 to 7 tons of oil on average per hectare. So if you are able to bring those yields up, double them, for example, you will make a big difference with regards to addressing that big food gap. So we work with oil palm. And essentially, it's a difficult crop to work with because it's large. It has a long gestation time. Breeding time is more than 10 years. Essentially, it is difficult to work on because of the fact that it grows taller. And the targets that we have for improvement are obviously oil yield, but also climate resilience and disease tolerance. So the question would be, are we able to produce more from less or from the same piece of land? And I hope that in this talk, I'll be able to convince you that it can be done by using the right kind of technology and the right approaches. So I'd like to introduce you to a bit of plant breeding, right? So in order for plant breeding is a complex undertaking, right? And there are, let's say, many tools that have been developed over many years that allow plant breeders to be more efficient and effective. But what I like to talk about is mucins genomic selection, right? Which is the technology that we are going to talk about and we have used for improving oil palm. So this technology essentially, or this idea, uses sequences across the whole genome as a means to make predictions or make selections. So if I look at the overall performance of what we have done at SD Guthrie or Sime Darby Plantation, from sequencing the oil palm genome in 2009, we were then able to actually take that knowledge from bench work and be able to actually convert it into a usable tool that could be field planted in 2016, which is in 7 years. And then after that, through steps of validation and so on and so forth, we are now actually planting large areas with these kinds of materials. So this is the oil palm genome that is basically an improved version of the genome that was published in 2013 by Malaysian Palm Oil Board. And this is about 89% better assembly. And it's in the public domain, and essentially it's put in the public domain to actually help other scientists to use this information to also innovate. So some of the products that we produced from this knowledge is basically a tool to ensure seed purity. We've developed a virtual breeding system. That means we can do predictions through a computer for accelerating breeding. And we are able to introgress in traits from wild relatives in a more timely manner. So let me tell you how we went about actually doing this project. So initially we sequenced about 230 palms of diverse origins. And from these 230 palms, we actually identified 6 million SNPs, or single nucleotide polymorphisms of high quality. And we selected 200,000 that were evenly distributed across the whole genome to be used as a tool to study over 10,000 oil palms with full data sets. In doing so, we've actually then been able to correlate some of those SNPs with the yield traits. And we have developed a mathematical model from deep learning and machine learning methodologies that allows us to do fairly good predictions on yield performance based on the DNA sequence or based on the profile of DNA that these palms have. And we've managed to actually reduce the number of SNPs that we use to about 1,000 to make it a bit more cost effective. So in the first project, we genotyped over 80,000 palms. And you can see, I mean, this is a huge operation, so it uses robots and stuff like that. You can see that these seedlings all have a barcode, and those barcodes actually relate to the genetic profiles of these materials, right? So this is the methodology that was used. So on the left is our first version of Genome Select. And essentially what it does is that we genotype all the palms and then only plant up those that have superior performance, right? So the top 30% are planted. This is a very expensive and time-consuming methodology. So from our learnings from the first project that we've done, we actually improved on it by being able to do predictions on the parental materials. So we can predict which parents would be able to combine to produce the best progeny. And then those are field planted. And of course, that requires a lot less effort to be able to undertake. So I think you're most probably aware that genotype alone is not sufficient to deliver the performance. There's an interaction with the environment, right? And it is a G by E effect. And sometimes it's somewhere between 40 to 50% of the genotype that is responsible for the performance that you see. But in some cases, the environment makes a bigger impact on the performance. So with this in mind, we actually planted the next generation materials in various sites to actually study and to see whether our predictions were any way accurate or not. And luckily, we have plantations or estates across the whole of Malaysia, Peninsular Malaysia, as well as Sabah and Sarawak. So we are able to actually do this. So how does reality compare with predictions? So firstly, this is what it looked like in 2016 and 2018 and last year, right? So you can see lots of bunches and nice looking cut fruit. So this is reality check. So the graph on the top essentially shows the first planting that we've done in 2016, and the yields are very good, and basically tracking on our predictions, in fact maybe slightly better than our predictions. And the table at the bottom essentially is the 2018 planting, so 2 years of data from different sites planted across Peninsular Malaysia and Sabah and Sarawak. And we can see, if you look at oil per hectare, that it looks very favorable. All of it actually is delivering higher yields than our controls, which are our current best materials. So this looks very interesting and exciting. However, most probably need to have another 2 years of data before we can definitively say that this has worked as well as we expected. So what to expect in the future? Well, definitely we're going to go for increased yields, ease of harvestability, meaning to say making palm shorter and easier to harvest, climate resilience, that means more tolerance to drought, and of course disease tolerance. So in conclusion, innovation is key to actually addressing many of these issues. We have to keep in mind scalability of that technology, so the technology needs to be able to be scaled, and that we need to actually deploy the relevant technology to make the biggest impact at the right points in your supply chain. So this will not be possible without these people. The slide at the top left are our board of directors as well as our senior management. And the other slides are our scientists who've done the work as well as our scientific advisors. I hope you enjoyed this talk. Thank you very much.